Hi, everybody. Welcome to Q&A with A&V, the last one for 2021. I am Vincent Racaniello, and joining me tonight from New York, Amy Rosenfeld.
Vincent, how are you today?
I'm very well, thank you. You look very spiffy with your shirt and dark sweater, your Cartier watch.
Yeah, and my Mickey Moto pearls.
I don't see any
Although,
pearls. Or are they? Oh, in your ears. Oh, okay, covered by the headset. Very good. Such a stylish scientist.
yeah, I
Stylish.
am.
I can't
I
even
am.
talk. Let's see, what do we have tonight? 244 so far. So
I'm
let's
sure we'll
dive
get
in.
more because everybody wants to go out for New Year's Eve. Are you going out for New Year's Eve?
Pff, what are you, crazy? I haven't been out for New Year's Eve in decades.
Do you have people over for New Year's
No,
Eve?
no.
Are
We
you
haven't
having
had
big?
people over in 10, like 5, 6, 7, 8 years. No, I don't. I'm antisocial now.
You're anti-social. Oh, okay. Are you having? Are you going to watch the ball drop?
No, I don't want. I
That's
hate
too
that. I
late.
hate that
That's
so
too
much.
late for me.
So already today in New York, they're putting up barricades all over the place, right? Because I guess the incubator is not too far from Times Square, right?
Yeah, you're less than 20. You're less than 15 blocks away.
Yeah, they were putting up barricades all over, so I wouldn't get near that. That's what is New Year's Eve? Friday,
Friday.
right? Now,
Yeah, you're not coming back until Monday.
I dislike very much the dropping of the ball. The best New Year's Eve I had once was the first year we had a kid, and we both went to sleep at 9 p.m. because we were exhausted from the kid being up all night, and to wake up the next day. Oh, Happy New Year! Good. Let's move on with our lives. Sister Woman says, I am not a scientist. I've never had a biology class. This show is way over my head, but has helped me cope. Thank you. Well, there you go. There's always a silver lining, right?
Of course.
I appreciate that.
It's good.
Appreciate it.
Good.
Let's see. Here's a joke. I have a joke for you. What is the most indecisive virus? That's good. This is Colopy Kittens, who's yes, in school, let's say.
She's the high schooler whose person told her she was going to become a ribosome or something.
I think so, yeah. What's the
I
most
don't know.
indecisive virus? I don't know, maybe a retrovirus because it can't decide whether it's RNA or DNA.
I don't know. Maybe she'll tell us in a minute.
I'm
She'll
hoping.
get,
I'm hoping. But it'll take a while to get to that because I'm delayed in the comments here. And we have tonight Mr. Ozzycam. We have Les, welcome. We have Frank. We have Steph. I don't see
Vaccinated
Vanity.
person?
I don't see Vanity. She might be flying.
No, she came back early this morning. She might be sleeping.
She did actually show up at the towards the middle of the Virology live.
Oh,
Yeah.
the
I just
last one.
the last one, although I've agreed to give a postscript in
What?
January. I'll do the viral ecology session. Yeah.
What is that? When is that?
I will. I want to do it before I start the Columbia course, right? Not probably not next week. Do you think I should do it Monday, or is that too soon?
No. If you're ready, Monday would be fine. Because then it's grant writing season. And I have to practice my talks and stuff. And I found out Ian's going on vacation at the end of the month.
All right. So
Also good.
Patricia and I have been chatting about antibody tests. Even Daniel wants tests. So how can we, U.S., the people, know our antibody levels?
Why do
You
you
can.
need to know what your antibody level is? I don't
Yeah.
think Daniel wants to know the antibody level. He just wants to know plus yes or no. He doesn't care what the level is
No.
because
So
the level
antibodies.
doesn't mean anything.
Antibodies are not good to. If you want to know if you're infected, you take an antigen or a nucleic acid amplification test, right? Because
Right.
the antibodies will be positive for a couple of weeks, and they won't tell you much. Either all they will tell you is that you have antibodies or you don't. We don't know what the levels mean, right, Amy, in terms of protection against disease.
Yeah, I know. I just had this conversation with the family members of mine who tested positive. And then her sister decided that she was the virologist. And I said, really? That's very interesting because I didn't see you at graduate school with me.
That's a good one. I like that. Great American Road Trip. There he is with that mug. Wow, we saw that last time.
Oh, we've seen him before? He's a regular?
Yes. Well, I recognize that mug. Look at that.
Well, that's huge. Is it beer or root beer?
Oh, I'm sure it's beer.
Wow.
Anyway, he said, thank you for your support of the incubator and science education. On day 8, testing positive on rampant antigens. Okay. Haven't had symptoms since day 3. Mild cold-like, three jabs in the original strain. When am I not contagious to be around elderly parents? No good guidance.
I think he's not contagious.
I think if you're testing negative, you're good to go, right?
Yeah, but he tested positive. He says he tested positive on day 8, which seems extreme.
Day 8. Oh, he's not negative yet. I would say that's going to be the extent of your positivity. Okay.
Seems extreme.
That's what Daniel is saying. Like anywhere from 2 to 8 days, 7 days after you'll be positive. So if you want to be careful, just take another test a day later, and I bet it's negative.
Yeah, I would think so.
So there you go. Hey, this is good. Thanks, mods, for keeping the pathogens out of the chat. Are they zoonotic?
Yes.
Rates have exploded here in Ireland. We've maxed out on testing, etc. We're watching ICUs. Hopefully, previous vaccines and previous infections will play a role in keeping, what's that, deaths down or hospitalizations down? They will.
Yeah, for sure.
So, Amy, did you see all these reports out of South Africa where that Omicron is a mild, and it turns out they think it's because those people were vaccinated.
No way. Really? Wouldn't have guessed. But yes, I told you that like 2 weeks ago.
I know. I'm making a little dramatic scene,
I know. And I then couldn't understand the whole thing about Scott Gottlieb and stuff. But who would have guessed a vaccine would make a disease less severe? Oh my God.
And so tell me if you agree with me, Amy. Eventually, everyone in the world will either be infected or vaccinated, and then most infections will be mild, and then it'll be another common cold, right? You think?
I agree with the first part. I don't think we'll ever consider it a common cold in our lifetime. I think everybody will. I think it will become a childhood vaccination, just like polio and measles. And it's just because we were so unprepared for it to begin with that we, we're going to overcompensate. But yeah.
So I've learned now, when I have a hypothesis, I first say to Amy, let me know if you agree with this or not, instead of just saying it. And it goes over a little better.
It's good.
Well, I only tried once, so you'll have to try it again.
It's good.
So Don wants to know, if you get monoclonals after testing positive, does it blunt your natural immunity or cause you to have a lesser T-cell response?
No.
Because if you tested positive, the virus has reproduced in you, right?
Right. It's probably a supplement, right?
To what? Vaccination?
To natural immunity. Monoclonals is probably as a supplement to natural immunity. Probably.
Well, it's just in helping tamp down the virus loads, you mean, right?
Right.
Yeah.
But it's not going to make a lesser T-cell response.
No.
I don't think.
Does Racaniello mean anything in Italian?
Yeah, you told me it does.
What does it mean?
I don't remember. It's not my last name. But you told
me the
me
Racaniello family was a prominent family of Umbrian origin. Now, right there, it's not related to me because I'm not prominent. Which
yes, you are.
In the 1400s, there's actually a Wikipedia page for Racaniello.
You have your own crest.
And they have a coat of arms of the Racaniello family.
Yeah, that's
Isn't that funny? The coat of arms of the House of Racaniello, that's so funny, is composed of concentric circles in yellow and scorched earth. The family motto is, I possess a refined art that to hurt cruelly whoever does me evil. So that's not me. That is not me. I don't hurt cruelly whoever does me evil. Anyway, I don't think it... Agnello is lamb, or maybe it, and anello is ring or circle. That's as close as I got. But my family said never told me it meant anything. But I know Rosenfeld means something, right?
Yeah, means you live near a field of roses.
Roses, and feld is field. That's very cool. I like that.
Yeah.
Animal Party says, here in Rhode Island, we are testing thousands of asymptomatic people. And I would like to know why. Why are we doing this?
Well, are they vaccinated or unvaccinated?
I don't know. It's a good question, but I bet they're a mix of both, right? The unvaccinated, I could see where you would test to see if they're infected, right?
I don't know. I'm sure she'll give us more details.
All right, Feed Me says, got my first COVID vaccine on December 14. Never an anti-vaxxer, but took this long for scientific truth to emerge through the relentless waves of propaganda and mind control efforts by both sides. I'm
Very
sorry.
good.
I hope you don't feel that we're part of the mind control effort. We are just giving you the data, and you can make a decision.
At the end of the day, if they want to say I was part of the mind control effort and they got over it to get vaccinated, that's fine with me. I just care about the fact that they got vaccinated.
Oh, this is good. I learned how mRNA works and was soon in line to get my first jab in March. That's great.
That's great.
That's how it should work. You learn, you learn how something works, and you go for it. In Canada, the waiting list to get a PCR test is 1 week. Health Canada says only test if you have severe symptoms. Is that good advice, Amy? I think so, right?
It depends on whether or not you're vaccinated or not, how old you are, and what is a severe symptom when you're half dead.
No, I think it's before that, Amy. I think it's before
I don't
that.
know what a severe symptom means.
Everyone I know has the same mild symptoms and is 2x or 3x vax. So this is what that does. Vaccination gives you mild symptoms. We've been saying this for a year now, right, Amy?
Yes, I have. I'm tired. I want to move
And every
on.
time I see the CDC say something, I think Amy said that a year ago.
Yes, it's ridiculous.
I just
It's
found
like
a
Groundhog
reference.
Day.
When? That's when we started?
No, like I say something, then somebody says no, then they come back and they say yes. It's like Groundhog Day. You know, you keep repeating the same thing.
So Vanity Nutrition has arrived.
That's great.
And she brought a loaf of bread the other day when she came to the incubator.
Yeah, it was green olive bread. It was good.
Let me show you a picture of it. I know that this is not a cooking show, but...
Why? You don't show pictures of my cooking?
Do I have one on my Instagram feed?
I don't know.
Anyway, I will do a screen of the picture. I think Amy can't see it, but...
Yeah, I see it. I see you in the olive bread.
Kind of looks like a virus, right?
Yeah, that's what she wanted. She wanted initially to make an adenovirus, but I forgot what this one
So
was.
thank you, Vanity, for that.
That's lovely. Where are my cookies?
Do I have pictures of your cookies?
I'm sure.
I don't know. Let's look. I will take a quick look. iCloud. All right, let's take a quick look. And maybe you could answer a question while I am looking.
Oh, I don't do that. That's
Here,
ridiculous.
this is a synagogue question. I'm on the reopening committee for my synagogue. I vote we do all remote until Omicron starts to fade. Your thoughts?
Ridiculous. Go to synagogue, wear a mask, call it a day.
That's
You
Amy
should be
for
all
you.
you should be all vaccinated to begin with, so that's ridiculous. You should go and have your services and wear a mask and blah, blah, blah.
I don't have any recent pictures of your cookies. It's a shortcoming. I will have to remedy that next week.
Forget it. They'll be eaten. It's fine.
No, the ones at the incubator will still be there. Oh, I have a picture of your stolen.
Yeah, am I stolen?
Yeah. Let me see if I can show this. Hold on. Let's go to the next email. After a person recovers from COVID, is it possible that he gets infected with the virus that was already in his body?
What does that mean?
Can SARS-CoV-2 activate again if the immune system declines over time? So in other words, they never cleared the virus and it recrudesces. Okay. You know what I mean by that?
Yeah, I know. I've not seen any reports of recrudescence.
No, I don't think so. But you never know. I mean, with Ebola, we found people harboring it for a long time, right?
Well, I just said I've not seen any reports. I didn't say it wasn't possible. I just said I have not seen any reports.
Yeah. So we reserve the right to be skeptical, but it may change, yeah. But so far, there's no evidence, as Amy has said. All right, let me get this picture of Amy Stollen. She makes a killer Stollen. I had a piece at the incubator. Where is it?
I don't know.
Oh, no, that's a almond croissant. That's not yours.
No,
Right,
but I might be making them.
That is, where did it go? No, it's just, that is the croissant from the place across the street. What is the name of that?
La Pan Queen.
Here we go. Here we go. So here we go. Here is Amy Stollen.
With the homemade marzipan.
There you go.
No, that's not even the good one. That's the one you told me was dry.
Well, it's the one I have, but you can see the beautiful filling there. What's on top? What do you call that? Frosting?
Oh, it's a mixture of hot buttered rum and confectionery sugar.
All right, so Questella wants to know, why do I have no clue what a Stollen is? Is that a German dessert, Amy?
Yeah, it's a German-Austrian Christmas thing. And it's, yeah.
So
I didn't know what one was until you told me about, you sent your disaster last year.
Yeah, we used to have Stollens at Christmas. It's a sweet. You have with coffee. My friends from Stuytown report seeing the longest lines for testing since the pandemic onset. Yes, people got out of control this year. For the holidays, for travel, people started testing. At the incubator last week, outside, people were lining up around the block to get tested at kiosks, at CityMD. You know, a lot of travel, they wanted to make sure they were tested before going to their family. I can understand that. But it's really...
Stop testing.
Stop testing.
Especially if you're vaccinated. Stop testing.
Does saying live virus imply that a virus is alive? I like your name, Fuzzy Bean. No, it's not. You shouldn't say live virus. People say it all the time because it's easy, rather than saying infectious virus. Infectious has 3 syllables. Live is 1. People go for the easy out, right?
Pretty much.
You shouldn't say live. It should say infectious. When I tell my colleagues who say it all the time, they say, oh, who cares, Vincent? Only you care. What can I say to stuff like that? Only Amy gets it, and she understands mutation, infectious, all that stuff.
I'm even more stringent than you.
Yes, you are. As evidenced here on this stream.
I didn't deny it. I owned up to it. What more do you want me to do?
Yeah, that's fine. If per CDC, most shedding is
need
done in 5
a
days.
little beanie with a propeller around my head? I mean, we could get one, but it would look kind of silly.
If most shedding is done in 5 days, does this mean replication is complete at this point? So I would say it's done even sooner. That's why they changed the isolation to 5 days, right? I think it's shedding is done in 1 to 2 days, after, especially in a vaccinated person. Yeah. So that's it. Yeah, there are no virus particles to infect anyone, or they're very low, and you can't measure them, or if you could, they would be too low to transmit. COVID count for today, 400,000. Yeah, holidays, that'll do it. A lot of people still unvaccinated, and as you know, vaccines do not prevent infection. Oh, by the way, I have something for you, Amy. I was, we've had a few questions on why the human papillomavirus vaccine is sterilizing. So this is one of the human viral vaccines that will prevent infection. Most of them do not.
The only one that I know of.
Yes. So I found a paper applying lessons from human papillomavirus vaccines to vaccines against chlamydia. And they say the immunological basis of HPV, vaccine-induced protection is high titer, long-lasting antibody responses in the genital tract, which provides sterilizing immunity. These antibodies are elicited through parenteral, that's injection, administration of a subunit vaccine based on virus-like particles. The successful generation of high antibody levels is due to the multivalent structure of the virus-like particles that are used. You know, Amy, I get that, but I don't understand why the levels remain high. That's just, they're supposed to contract, right? I don't know the answer.
The truth is you're suggesting that that all of it contracts because it's IgG, right?
Yeah.
I don't know what the rate of contraction is for IgA, and if it's mucosal, it should be IgA. I don't know what the kinetics are.
So viable is another word you could say instead of infectious. Viable is still
Viable?
3 syllables. Yeah.
How is viable virus right? It means alive.
It's infectious. Does it mean alive?
It means alive.
All right, Amy says no. Amy says
Not good. Not good.
A&V, in North America, when will life go back to some sort of normal? Omicron is everywhere. Makes me feel like this will never end. Thank you. Amy.
So it should have gone back to normal for this holiday. Hopefully by the summer, by the 4th of July, it will be back to somewhat normal. But at the rate we're being run, most likely it'll still be a 5-year pandemic.
Okay, Taylor says, saw some real-world data from South Africa. All right, so Taylor, just be careful about real-world data. All right, these data
I don't know what real-world data means.
Coming out of hospitals, it's very suspect to confounding factors. 2 doses of Pfizer, 33% protection from infection, 70% from hospitalization. Less virulent, huh? I don't buy those numbers. I'm sorry. What do you think, Amy?
I don't buy the numbers, and I don't believe the conclusion of less virulence, because that implies that it's a viral phenotype or a viral thing that is controlling that phenotype, and it's not. It's the fact that you have pre-existing immunity that interrupts the replication and the reproduction of the virus at different steps, so that you don't have the ability to develop severe disease and stuff. So I don't like this idea of the less virulent, because it suggests that it's a virus thing, right? And it's not.
Let's wait for more data, Taylor. These early reports are almost always wrong. Has there ever been...
always wrong?
Always wrong, sorry.
I was waiting for one you could name that was, right?
Has there ever been a virus that mutated to become more transmissive over time or in any previous historical pandemics? No, not that I'm aware of. Are you aware of any, Amy?
No.
And I've always said this, that transmission could be a selection force, but I never saw it happen. And whether it's happened here, we don't know. The data are not in yet. We'll see.
But how are you going to measure transmissive force?
I knew that was coming next. You see, I got your number. Dude, I got your number.
I mean, is transmissive
it's hard.
Force... do I get out my lightsaber and go with like the force be with you kind of thing?
No, I totally get that. Measuring transmission is hard. It's very difficult to do. Do you have any advice for what to say to people quoting Robert Malone? I don't know who he is. Do you?
He's the guy who thinks he invented the mRNA vaccine.
Okay.
He's now an MD. I don't know. I don't know what he says.
Well, I mean, Drew Weissman and Kariko, what's her name?
I don't know what her last name is. Let's just put it to you this way, that there's a lot of fighting among...
Catalin Kariko. They got the prize from Columbia, the Horowitz Prize, which is considered a precursor to a Nobel. Those two, not Robert Malone. And so they know what's going on.
Well, it turns out that there's a lot of people who are trying to become part of get credit for it because like there's a lot of fighting about the patent for it all.
I'm sure. When money's
It was in
involved,
the journal.
people fight. Wall Street?
Yeah, it was a big
Well,
article.
I can't get to it. It's a
Well, I
paywall.
can, fine, I'll send you the article.
So, can you catch COVID from surfaces, Amy?
No.
I think not. We thought so in the beginning, so we did a lot of hygiene theater, but I don't think there's very much evidence for fomite. However, let me ask Amy this: can you catch rhinoviruses from surfaces?
Yes. That's why. Where's my beautiful slide that I made? I sent you this afternoon. That's why rhino doesn't go down.
Yeah, let's show that. That's a good idea. Let me pull up that email. Where are you?
No,
what's your name?
Text.
Yeah, it's a text. I know. Here we go.
You say email. How do I know that you know it's text, not email?
Wow, I misspeak a lot. I misspeak. No, that's not what I want.
I'm done with the stolen. That is too dry.
Can you see this now? No, the stolen.
I
have to get rid of the.
I'm done with it. It was too dry. It was the wrong recipe.
Where did the damn thing go? I found back to the picture. Let's do a. Do you see it now?
Yeah, make it bigger. People won't be able to read, but yeah.
Circulating respiratory viruses during the SARS-CoV-2 pandemic.
So it's from June 2020 all the way to basically the beginning of 2022. And the blue is rhino. Rhino/entero.
So it's this held at a steady level, right?
Pretty much.
Despite masking and distancing and so forth. That's your point, right?
Right.
And what is the orange?
The orange
coronavirus.
It's corona.
And some of these others have fizzled out and now are just returning, right?
Yeah, you can see that RSV was at 1.3% of the positive swabs. So like June, and then it went up to 12.5, and now it's gone down a bit to 7.1. But rhino was 4.5, so it was the predominant thing outside of the corona, because we were in a corona pandemic. And then it goes up to 30, and then it hovers at between like 22 and 30.
Okay. Are some viral epitopes denatured by inactivating agents?
Yes.
Yeah. That's why if you want to inactivate a virus with, say, formalin, like inactivated polio vaccine, you make sure you check that the protective epitopes are still there. Nevertheless, some of them are inactivated, right, Amy? Because there's an antigenic difference between IPV and inactivated poliovirus.
Yeah, for sure.
Quebec has announced that healthcare staff, vaccinated or not, that test positive and asymptomatic will be required to keep working. Is this a way to get herd immunity, or what do you think about that?
I think it might be fine if they were vaccinated, depending on what their clientele is. If they're unvaccinated, I'm probably not good with that.
I think this is an effort to try and keep staff upright, but could have dire consequences. I assume if they get symptomatic, they will be sent home, no?
Yeah, I don't know that I would say that an unvaccinated who tested positive is asymptomatic is not shedding. I don't think that there's any relationship to the amount of virus reproduction and symptoms.
Will original antigenic sin interfere with the effectiveness of a pan-coronavirus vaccine?
No.
I don't think so. I think this is an influenza issue mostly, right?
Yeah, or maybe dengue.
Yeah, there's no evidence that it's going to be an issue with coronaviruses. All right, Vincent and Amy, Omicron and Syrian hamsters. Why is knowing host range important?
Because it's a zoonotic virus, so you want to know if it can go back and forth between humans and animals and change. And it helps if you had another animal model, right? An endogenous animal model.
Yeah, so a model would be good because other variants of SARS-CoV-2 can reproduce in hamsters. But also you want to know what the changes are doing and the potential impact on infecting animals in the wild, right?
Right.
I mean, what if it turns out that Omicron doesn't reproduce in deer? That would be good, right?
Well, it's not killing the deer, is it?
No, it's not. But then say there's a ancestral variant in deer, and then that's been outcompeted by others. So that would suggest that if that virus came back into people, it wouldn't be an issue. But that's all these things you need to know.
I don't know if I would say that. I don't know if I would agree with that.
I forgot to say, what do you think about this?
I didn't say anything. I just said I don't know that
it's true. I understand. I'm walking on thin ice. I got it. Why should I bother with a booster if Omicron is like a flu, given that I'm double vaccinated? Well, it is. We didn't know it was like a flu, but it is in vaccinated people. We don't know what the virus would do in unvaccinated people yet.
But he's not unvaccinated. He's only concerned about him. And we're not concerned about the unvaccinated because they're rolling the dice and taking a risk. So if they're happy rolling the dice and taking a risk, that's fine with me. They're not me.
Well, the thing is that after 2 doses, and we did this paper on TWIV a couple of weeks ago, your antibodies don't neutralize Omicron very well, but they do after a 3rd dose, and so that's what convinced me.
Right,
but I agree that we don't know the relationship of that to disease. I totally understand, but
But the truth is you didn't measure whether or not non-neutralizing in your tissue culture dish antibodies have any role in protecting against Omicron, and you negated the T cells. So it's not really clear, and people have made way too much out of this binding of neutralizing antibodies.
Yes, for sure.
So,
but a lot of this 63 bits is in retrospect. So, I mean, it may be that in a month we see that, we don't need a 3rd shot for Omicron. But what about what's next, right?
No, the truth is most likely you're never going to need a 3rd shot, right? You're just going to need a boost like every 10 years, right?
Well, I mean, look, the breath after the 3rd dose is what convinced me. Whether you need that or not is another question, but I thought that was useful to have. But I understand that not everyone might. Are we likely to be playing whack-a-mole with new variants until we get a pan-coronavirus vaccine?
What's whack-a-mole?
Oh, Amy, it's the game at the amusement park where you have a mallet and a mole sticks its head out of the thing and you hit it, and then they're all holes in the thing, and then another one sticks its head out and you keep hitting them down. You get points for each one. It's called whack-a-mole.
Did you play this?
Yeah, I did. I was really good at it. I had good reflexes when I was young. Maybe I still do. I don't know. I should go play whack-a-mole.
Maybe we can just go to Seaside and we could get...
Well, that's where I played at the shore, at the Jersey Shore. Anyway, no, we don't need a pan-corona vaccine. The ancestral vaccine that we are using today still protects very well against serious disease and hospitalization. Yeah, you're going to get a cold.
Wait a minute. We don't need a pan-corona vaccine? I think we do. It's a separate issue.
All right, you want a pan-corona, that's fine. That's fine.
We need a pan-corona to protect us from the next...
From the next one, not from a variant of SARS-CoV-2.
Right. Not from this variant. But so I think, there's 2 separate issues.
The claim that human coronavirus OC43 caused the 1889 Russian flu pandemic is made a lot. Any actual evidence supporting this and any reason to think COVID-2 will ever be as inconsequential as the 4 other seasonal human coronaviruses. Amy, have you seen any evidence that the 1889 Russian flu was caused by OC43? I've seen none. I mean, it's we have no samples from then, so we really can't prove it at this point, right?
Well, how are you going to dig up the bodies?
Well, we did that with 1918 influenza bodies, right?
Right, but it was really hard to piece it all together.
Well, someone has to have the will to try and dig up some of those 1889 bodies, but not going to be much of them left. Yeah, exactly. You do need lung tissue or upper tract tissue, right?
Well, good luck to you.
And
yes, that works out.
I think that the COVID-2 will eventually be a common cold once most of the population is immune. That's what we've been saying for a long time. Yeah, it's now getting some press, but we said it over a year ago. And I have to say that Ralph Barrick first mentioned it when he was on TWIV the first time, February 2020. He thought it would do that. So, but no one listens to TWIV.
I don't know. You got 904 views now. Somebody's listening to
I'm
Twitch.
sorry. I don't mean nobody. It's just the vast majority of the world, but that's fine. Why does the FDA want 55 years to process Freedom of Information Act requests over vaccine data? I have no idea. This out of my wheelhouse. What about you?
I don't do legal stuff.
You sometimes have comments on these corporate mumbo jumbo stuff. You're broader than I am in this. No, you don't know. Okay, I'm sorry, Taylor. Maybe someone else here has.
I'm not even that interested.
All right, so someone said, can you use evolutionary distance to hypothesize that 1889 was caused by HCV? We don't have any sequence from 1889. You could back calculate from current HCOV sequences, and you would just find out that it's already been done. We know roughly when it spilled over from, I think, cows a few hundred years ago or 100 years ago. Maybe, that's part of the evidence that it was 1889. I think it's coincident with that, but that's not good enough. Ronald says, thank you for your contribution to science education at the incubator. People were saying before in the pre-show that they really like the name incubator, and I said you had thought it up. I gave you proper credit.
Oh, thank you. Yeah, I just you needed something like the factory.
So I have to say that Anthony, who's a listener from here in New Jersey, he's been a listener for many years. He emailed me once. He said, you need an Andy Warhol type factory name. So I ran that by Amy, and she immediately said incubator.
Yeah.
Sharp as a tack.
Yep. And saying the virus is more fit is not equivalent to saying it's more contagious.
No. I know this is a nuance, but it's not the same. It's just, it displaces, fitness means it displaces other variants. And why it does that, we don't know. I am scheduled for a Moderna booster tomorrow. I have a mild cold, runny nose, absolutely fine. I have no idea if it's COVID. Should I postpone the shot?
I think Daniel would say you should. I think Daniel would say you should do a rapid antigen test before, because you don't want to, even though you're probably going to go wear a mask, you just don't want to get the person who's the healthcare worker infected. You don't want to put them at risk.
All right. So Doug says the Freedom of Information request for 1/3 of a million pages. They have limited staff to review and redact confidential information, so it takes many years to deliver it. Okay.
Okay. I don't know why you need 1/3 of a million pages. Do you know how many pages that is?
300,000 pages. It's not that much. I have 300,000 pages in my lab. Well, maybe not that much.
That's over 25 years.
How can Walter Reed's ferritin VLP vaccine achieve such breadth of protection using only one kind of spike?
Same way as the Moderna spike and Pfizer spike.
There's the Moderna is one kind of spike, and you can get breadth as long as you space out the doses properly and allow for somatic hypermutation. I don't think that's anything unusual from the ancestral vaccine that we've been using. Do all SARS-CoV-2 variants have the same number of spike protein, or does it vary?
Probably varies a bit, but in the ballpark. It's not like it went from 10 to like 400. It might go 10, 12, 8. No.
Yeah. Let's see. Costello said, my New Year's is 28 hours at home doing every single time zone and learning something about somewhere in every time zone. That's a very nice New Year's, actually.
That's cool.
What country has the first New Year's?
The country that's closest to the date line. Is that like Fiji?
I don't know. Someone's going to tell us.
Okay.
Now. Bottom line, does this Omicron variant appear to be milder? As Amy said earlier, you can't tell if it's the virus or it's simply the level of immunity in the population. We don't know. We may never know. I
And
got
all
these.
for that matter.
I got these at Amazon. These frames. They have they're very special. Yeah, right. I'm going to tell you exactly the name of them. Glasses. I can search my previous orders. It's a company called Didinsky. Dinsky. Okay? They come in different colors. I bought what did I buy? Purple ones, but by mistake I bought clear glass. These are a little bit of magnification so I can see the computer screen without tilting my head back. I'm sorry to give all this information, Amy.
No, it's fine. I'm good.
How big of escape risk are we taking introducing antivirals one by one versus waiting until we could have a cocktail? So here's where I differ with Amy on this. I think it's a big problem. I think we're going to have resistance to both. Amy thinks we will have resistance to the protease inhibitor, but not the molnupiravir, the chain terminator. Is that correct? It's not a chain terminator. It's a mutagen, right? You don't think we're going to have resistance to that, right?
I don't know that I said that, but if I said that, I guess that's what I said.
Anyway, I think it's a problem, but we can't test them. We can't wait to use them as cocktails. That would take another 6 months. So
I don't know. I was for that. Screwed it up the first time. What's the problem now?
All right, Taylor. So an infectious disease doctor stated, who is not a virologist maybe, that Omicron is now the second most contagious virus on the planet. I don't believe any of those numbers. As Amy will say, R0, what would you call it?
The made-up number?
R0 is made up of human behavior and virus behavior, and there's
No,
no
it's
way.
a made-up number.
There's no way.
It's made-up number. Hold the envelope up to your head. Made-up number.
These calculations are BS. In fact, anyone will tell you that you cannot calculate R0 after the outbreak has begun and there's population immunity, blah, blah, blah. It's a different number. It's an RE or RT. But even then, it's so squishy. I'm sorry. This is
the
not
whole concept is wrong. It's not an intrinsic property of the virus. You're measuring how crowded people are and how much they are able to wear their mask. It's just like, do you know that the R0 of EV71 in the daycare center in Taiwan is 32?
Really, what is it in New York?
Well, there is no EV71 in New York.
Yeah, that's true.
So it's
That's
0.
ridiculous. So that would not make
32.
Omicron. That would not make Omicron the second most contagious virus. It would be, the most would be EV. So that's the point Amy is making. EV71 would be the most contagious virus in Taiwan in daycare centers. Do you get it? It's all about human behavior that runs these numbers, not the virus. And people don't get this. They think this is a constant describing a viral property and that the variants are changing. We've been saying this for a year. And, you know, a lot of you still don't get it. Just wait another year. You'll see.
I'm telling you, it's because 3-year-olds don't like to use Kleenex. That's why.
Yeah, the 3-year-old can change the R0. And here's another thing, Taylor. You can bring the R0 down below 1. That's how you stop an outbreak.
No, you
really?
stop an outbreak. No, you stop an outbreak by having less than 1 per 100,000 people. That's the number.
I understand what you're saying, but many people will say if you bring the R0 down below 1, you can stop an outbreak. And how can that be that you can actually, how can you change the R0 if you think it's a physical, intrinsic property of the virus? It's ridiculous. Ridiculous.
Yeah, it's called stay in your house without the 3-year-old.
So the infectious disease doctor is simply repeating information that he or she has heard elsewhere without thinking about it clearly. And I don't mean to insult them, but this is ridiculous to say this. Unfortunately, most of the planet believes this.
Unfortunately, most of the planet is non-thinking.
Well, I don't know about that, Amy. You know, our people here tonight are thinking. What viruses are transmitted by fomites? Rhinoviruses. I would think Ebola viruses also, the contaminated blankets and so forth.
Smallpox.
Smallpox, yes, for sure. Nipah virus.
Isn't that what we did in the Revolutionary War? We gave all the Indians the smallpox, or was that the French and Indian War?
No, Native Americans, Amy. Native Americans.
Okay, Native Americans.
Or if you're in Canada, it's First Nation, I think, right? What's up with this new Cobra Vax in Texas?
Oh, that is Peter Hotez. He's got approval to distribute it in India.
Yeah, developed by the Indian biopharmaceutical firm Biological E Limited, Baylor College of Medicine. Protein subunit vaccine. I haven't seen any data. Have you, Amy?
No.
Thank you, Elizabeth, for your contribution. Really appreciate your support of the incubator. What's the most recent baking project? Wow. Should I show him a picture of this thing you bake? What's the name of it? Tell me the name.
It's the 8-layer Russian honey cake.
Russian honey cake, 8-layer. So,
it's such a
oh my God, look at this. Look at this. I'm going to show you a picture of this. Where did it go? It's going to be small, but there it is. Look at that.
Yeah.
So I showed her this, and I said, "Can you imagine this?" It's going away. There's a video there. Sorry, and then the next week she brought it in. Oh my gosh!
Well, you're gonna cut it tomorrow with Aiden, right?
And I brought it home, and we're going to eat it. Yeah, she is an amazing baker. I have to say, folks, she's really got talent. And I don't know. Do you think it's because of your science skills?
Yeah, could be.
Is an N95 that's not fit tested with the machine safe enough? Will an air take the easiest route through the gaps? As my understanding is 95s have to be fit tested to work optimally, yes.
I don't know.
According to Daniel, he did a little review of masks. Are you going to leave us soon?
Yeah, it's 8:45 and I need to put overlay on, so like 2 more questions. And some guy complained about how I insulted the whole world. I didn't insult the whole world.
But you should understand that when Amy says that she means it's a generic way of her saying people need to think more.
Yeah, it's just frustrating. Oh, Vanity Nutrition corrected us. It's a Medovik cake.
A what?
Medovik. Medovik cake.
Well, it said I searched for Russian 8-layer cake, and that's what came up. Okay, yeah, it's fine.
Well, you're going to take a picture of the layers when you cut it with this.
Yeah, when I cut it, right? I'll cut it. I'll show you guys next week on the live stream.
Well, I want to see it because this is the only one that I've made.
Yeah.
I've never made it before, and I might not make it again because it's way too big for one person.
U.S. approved molnupiravir. India did too. Multiple pharma are going to produce it. Do you agree with concern that the drug will lead to dangerous variants? No, I don't think it will lead to dangerous variants. It will lead to resistance most likely, but not dangerous variants. Ditching, thank you for your contribution to the incubator. Amy, do you know when the plant-based vaccine will be approved?
No.
Well, they're at the moment they are looking for approval. They have the data, so I don't know. Maybe next year, sometime early next year. But I don't know anything for sure.
All right, 3 more and then I'm done.
What do you think of revised CDC guidelines of isolation of 5 days for asymptomatic positive individual? What do you think of that, Amy?
Depends on whether or not they're vaccinated or not. If they're vaccinated, I don't agree with it. No need to isolate. If they're unvaccinated, yes.
Why are viruses so easily destroyed by soap? Not all of them. The enveloped ones tend to be, but polio is not destroyed by soap, right, Amy?
No, not at all.
Yeah, you can put polio in soap and it will laugh at you. If a virus could laugh, right? So It depends on the virus.
Well, soap disrupts the lipids. That's what soap is to do. Why would you think it's any different than when you put fat in or oil in soap? Soapy water.
Yeah. Okay, so we got the joke. It was retrovirus is the most indecisive virus. I got it, Amy. I'm clever, aren't I?
Yes.
Vincent and Amy, how do tests distinguish between antibodies from vaccines and from natural infection? Go ahead, Amy.
Well, some tests look for antibodies against nucleic capsid, which you would only get if you were naturally infected because all the vaccines only produce spike.
Right. One more.
One more. Then it's time to go.
Will I need another booster despite being vaxxed with one J&J and a Moderna to be protected from severe disease? Got vaxxed in April and boosted in mid-November.
Should be fine. But
Oh.
they're probably going to make it that full vaccination means 3 doses. I can tell you that that's what they're doing in New York State. That I've already seen things that have said in order to be considered fully vaccinated, you have to have 3 doses.
Could you comment on the super antigenic motif? We had a TWIV on this, remember?
Mm-hmm.
Moshe, or DT, and the lady from Pitt, whose name I don't remember, but they presented this nice story. So a super antigen is one that can, activate a certain kind of T cell, right? And they become overrepresented. And this, there's a super antigen. And what are the consequences for that, Amy? Of that?
I don't know.
Having a super antigen. It's not clear what the...
Yeah, I don't think he was clear.
Yeah, but a super antigen is found. So super antigens have been found in other pathogenic proteins or virus proteins of pathogens, and they can often, cause this biased T cell response, which may not be good. So I'm not sure that he thought that it was playing a role in the multi-system inflammatory disease of kids, remember?
Yeah.
Right? So you get an unbalanced T cell response.
Yeah.
Anyway,
right.
search super antigen and TWIV and listen to him, that episode. It's really good. All right, Amy, thank you so much for joining.
Well, Happy New Year. I guess I'll talk to you tomorrow.
Yeah, I'll talk to you tomorrow.
Maybe not. And you're at 994, so I'm sure you'll be over 1,000 by the time you go to leave. All right. Happy New Year.
All right, I'll talk to you tomorrow. Bye-bye.
Bye.
Say Happy New Year to your fans.
I did. I said Happy New Year to everyone.
Okay. Bye-bye.
Hey.
All right. No, I wanted to show you guys my background is back. I got rid of the damn furniture from the flood. And so now you can see my, that's the other finger. My viruses up there on the shelf. And, of course, the red guitar, which doesn't get used. Oh, I forgot to put my light on back there. I have to turn it on. It's just inappropriate not to have my light. I'll be right back. Now, there's another thing I want to show you. Now, you see that model on the shelf there. I've shown it a couple of times, poliovirus, the magenta and the blue. Poliovirus and its receptor. And that is a model I had made, and it's also a model that's on my cell phone. Well, it's covered by my vaccine stickers. But when I had it made, the guy sent me a couple of gifts. And look at this. This is very funny. This is a Christmas tree ornament. It's got a TWIV on it, right? And this is, there's the hook. I didn't put it on the tree this year. I should have. But this is one receptor bound to the virus particle, all right? So I'm a total nerd about viruses. I would put this on my tree. I have like 5 of these. Isn't that cool? TWIV. So he sent this to me unsolicited because he's a TWIV fan. How cool is that? All right, let's get back to some questions. We have about another hour or so. Let's see. What would be a safe positivity rate or case number to go back to crowded bars and restaurants again? Well, I have no problem going back to crowded bars and restaurants already. I went to lunch today, and to eat, you need to take your mask off. Why? Because vaccines work. I understand there's maybe a 10% chance that I might end up in the hospital. I could die. I understand the vaccines are not 100%. I'm willing to take that chance. I'm healthy. I have no comorbidities. And I do think that the people who get very sick have other issues, mostly other health issues. But Amy always says one case per 100,000 population would be a really safe number. So I'll go with that. Is there technology for a vaccine for SARS-2 that would protect for longer, or do the changes in viral behavior preclude this? Yes, we could make, well, look, so these broadly acting vaccines that would take any coronavirus that came at us from an animal reservoir. Those would be great to have. They're a little harder to make because how do you do it? What part of the virus do you target, right? So right now we're using the host spike, and it may be that using a smaller part of the spike might be better. So it's not a new technology. It's just a different approach to the immunogen, I would say. And people are also working on that for influenza, and I have no doubt that that will work in a number of years. I don't know how long it will take. If your affinity maturation occurs for months, are your B and T cells still holding on to some antigen? So this is the theory, yes, in the germinal centers. And eventually the germinal centers go away, right? And then they come back when the antigen comes back. But the idea is that there is some antigen stuck in there for these months. Yeah. And so depending on the antigen, maybe it could be sticky. Some could be stickier than others. So that's a really good point, yeah. Nice preprint from Sete at El Sete. Will be on TWIV in January, early January. So look for him. We'll talk about that for sure. My cousin's a nurse in New York, had second dose and booster, seriously ill with COVID, was exposed many times prior to vaccination, didn't get COVID, high viral load exposure. Yes, well, of course, as I said just now, the vaccines are not 100%. They're 90-ish% protective against hospitalization and severe disease. So, in a big population, that's a lot of people. And so even at 90%, it's a great vaccine. We're really very lucky to have this. But there are going to be people that get sick. They are small numbers, and I'm sorry, it's your cousin, but we cannot let this make people say, oh, the vaccines don't work, I'm not taking them. It's not the case. They do work most of the time. If the nucleic capsid is packed inside the virion, how do we form circulating antibodies to that protein? Well, that's a great question, Lamont. And that is because when the virus is inside the cell. So the virus enters a cell and it begins to make proteins. It makes individual proteins, and inside the cell, they're there before they get packed into the virus particle, right? And so those can be chopped up by the cell and presented and lead to the production of antibodies. Or antigen-presenting cells like dendritic cells and macrophages can scavenge virus particles. These proteins can be released from cells that die, and the antigen-presenting cells can pick them up. And then they will process them and present them to T cells in the lymph node and generate antibodies. So to make the antibodies, there's a part where those nucleic capsids are not always on the inside of the particle. Good question. Yeah. Well, I'm not sure what you mean by what was a favorite title in the virology world? A title. What do you mean by a title? A book? An article? I'm not sure. How can the intrinsic virulence of a virus be measured, quantified, completely independent of the host immune response? So measuring virulence is very difficult because you do it in animals in the lab, right? You cannot do it in people. I mean, we get some idea that, Ebola virus, people use the case fatality ratio as a measure of virulence, right? Ebola virus can kill up to 90% of infected people. The problem with that is, in the right country, Ebola virus won't kill anyone because we know how to take care of the infection. There were a handful of Ebola virus infections in the U.S. They survived. So even that number is squishy, like the R0 is squishy. All right, so that's hard to do in people, but you can't infect people, so there's no way that you can do it carefully. So you have to use animal models. And then you could make animals without the immune system, but then most viruses will just be incredibly lethal without the immune system. So it's not fair to take away the immune system because you'd like to know in the presence of an active immune response is the ability of this virus to cause disease. I can tell you that if you take out different parts of the immune response, if you take out, for example, innate immunity, well, many viruses kill all of the hosts that you infect. So it's hard to do, and that's why we don't have any quantification of virulence yet, because we don't have good animal models for this particular virus. Can you explain in layman terms why additional boosters will not be needed? Why would we not need a booster every year like the flu shot? Okay, so the booster, the third dose, turns out to fix the fact that the first 2 doses of vaccine, at least here in the U.S., were given too closely. 3 to 4 weeks apart is not enough for the second dose to get the fully matured antibody response. Because when we give a booster 6 months later, now we have antibodies made that can neutralize any variant that you throw at the antibodies. So that tells me we don't need to boost every variant. Now, here's the caveat. The vaccines were originally tested to prevent disease. So in the vaccine trials, they immunized certain people. They gave certain people a placebo. And then they said, what kind of COVID do they get? COVID, any kind of COVID, mild, moderate, severe death. And they lumped them all together and they said, okay, a month after you get the vaccine, you're 94% protected against any kind of COVID. Then, as the months passed after the vaccination, that declined, and people freaked out because then people were getting infected as antibody levels declined. However, they were not getting severe disease. We still had 90 percentile protection against severe disease and death. 90% is not at 100, remember. But the protection against infection was declining. Well, that's normal. Most human vaccines, except the human papillomavirus vaccine, do not prevent infection. Neither does COVID. So if you want to prevent infection, then you boost everyone every year, because that pops your antibody levels up high and it will prevent you from getting infected. And that's what apparently they're doing in Israel. They want to give a 4th dose and then a 5th and then a 6th. I don't think that's necessary. It really depends on what is acceptable, right? Is a cold, a mild cold, and maybe in some cases a more severe but not life-threatening cold, is that acceptable? Or do you want to get boosted every year? I can tell you that some people will be boosted every year and others will not be. Now, the reason we do it for influenza is because the virus undergoes antigenic variation like the variants of SARS-CoV-2, except in that case, a small antigenic change in influenza can then result in an increase in severe influenza. So we're not seeing that with SARS-CoV-2. We are not seeing in fully vaccinated people an increase with every variant in more severe disease. I don't care what numbers you're looking at. We don't see it. We do with flu, and that's why we changed the vaccine. So I would guess, and I'm sorry for the long-winded answer, it's a good question, that if we start to see an increase in the general population, not just in over 65, an increase in severe disease in fully vaccinated people, then we will boost. And we'll do that as long as we have to. But if you just want to prevent infection, you're going to boost anyway. So that's a long-winded answer to your question. I hope that does it. All right, we have a quote by Christian Drosten. All right, let's see what he said. If you believe that you can train your immune system through an infection, you must also believe that you can train your digestion through a steak. Well, I don't really like this statement at all. I'm sorry. I mean, people think Drosten is a god, but not everything he says is great. And I know he's got millions of followers in Germany that adore him, but it doesn't say everything great. He thought the study out of Hong Kong on the replication of virus of Omicron in human cell explants was great, and I thought it was inconclusive. All right, so you get vaccinated to give you an experience with the virus, but one does not hurt you, and then you're immune to it. So that's training. And he's saying you shouldn't depend on an infection to get that kind of immunity. He makes the analogy with a steak, but that's not right. The problem is that infection can lead to severe disease, whereas the vaccination cannot. So I don't particularly like that at all. Why can't we correlate antibody levels with disease? Why is this not important? Well, it may be important, but antibodies are really easy to measure. You take blood, you separate out the cells, and you take what's left, you mix it with virus, and then you measure the infectivity. It's really easy to do that. You can do it in a day. And it's not necessarily clinically useful. It's an easy assay. There are other things that correlate with disease. For example, T cells. The other arm of this adaptive response. T cells are really important for protecting you against severe disease. So if the antibodies that you make are not quite neutralizing the virus and it gets hold, the T cells can still come and kill the infected cells because they are not, the epitopes, the proteins recognized by the T cells don't change in the variants anywhere near as much as the sequences recognized by antibodies. But it's really hard to measure T cells. So someone just pointed out Alessandro Sette's recent preprint. That's what they do. They measure T cell epitopes and they say there are some changed in the variants, but they're not going to make a difference in your protection against disease. Also, there are other kinds of immune activities that are not easy to measure that may correlate with protection. And that includes antibodies that are not necessarily neutralizing, but do other things to protect you against infection. And we did a paper on TWIV recently where they were testing respiratory syncytial virus vaccines in non-human primates. And they found that for some of them, protection correlated not with neutralizing antibody, but with other antibody functions. They're called FC because it's the other part of the antibody that's involved. We just did a paper on TWIV, which will drop this weekend, where they tested a Lassa virus vaccine, and protection in mice did not correlate with neutralizing antibodies. The mice didn't make neutralizing antibodies, yet they were fully protected by this experimental vaccine. So that's why we can't correlate antibody levels with disease, because we haven't done the right studies to say which antibody level protects you and which protects you against infection versus disease. And that's why with Omicron, it evades antibodies very well. Still, in vaccinated people, the infection is mild. Why? Probably because of some other kind of antibody or because of T cells. And please remember that when you read all the naysaying in the press. It doesn't seem to matter that much what antibodies you have. And then people say, oh, but I want to have antibodies to help me. Yeah, but maybe they're not so important as the variants are teaching us. Do we know if Omicron is more airborne than previous variants? It's exploding. No, I don't think it. I mean, someone just put up R0 values, and I think that's preposterous to calculate these R0s. As I said, you can't calculate an R0. It's a different number, and I think this is all driven by human behavior. Mostly unvaccinated people, but also vaccinated people, because they are out and about more than they were a year ago. They were masked and sheltering. A lot of unmasked people walking around. They're going to restaurants now. And they can be infected. So they test positive. They go for a test and they're positive, and they may not have any symptoms. Then there are still a lot of unvaccinated people out there. I am not convinced. And I know there's people on this stream that don't believe this. They think it's all the virus that's driving this. I'm not convinced. There's no data that suggests that. I think it's all driven by human behavior. I think perhaps there's a bit of immune evasion by Omicron, but as I said, you're protected against severe disease. So what difference does it make? Let this burn through multiple cycles, and eventually it'll be a common cold. How quickly will Delta be displaced, or will they co-circulate? Well, it's hard to know, because right now we're in a big wave that could potentially displace it in a few months, but the wave is not going to last a few months, right? It's going to probably be over, I don't know, at the end of January, February-ish. Although, students are going back to school in January, so that could keep it going. And remember, that's a lot of people moving around. That really drives it. Holidays, student movements, restaurants opening, theaters are open. People are back to doing things that they weren't doing a year ago. Please consider that when you talk about outbreaks and, big infection rates. Could a variant revert back to the original strain gene sequence? How would they? I doubt they would revert. I mean, it could be that there's some ancestral variant somewhere and we don't detect them, and then a variant arises that can't compete with that one, right? So we have a sequence, right? Alpha, beta, delta, Omicron. But maybe Omicron wouldn't compete with beta or alpha. We don't know. So I don't think it would revert, but it might be and it could be that these variants, these older variants, are in animals, and then they could come back into people. Who knows? Who knows? Is it true that lockdown measures have driven one of the influenza lineages into extinction? If so, is this a positive development, or will it just get replaced by something else? So I'm not aware of this. I don't know what lineage you mean. I find it unlikely that this would be the case. Now, the H2N2 lineage disappeared in 1968, was replaced by H3N2 and then by H1N1. So currently we have H3N2 and H1N1 circulating, and that's not just a lineage, that is a subtype, so that's pretty higher up. But H2N2, we don't know, we've not seen it, but it could be in birds and we haven't detected it, and that's a candidate for one coming back because there's very little population immunity. So the bottom line is, Roger, if you extinct the lineage, you're not going to have as much population immunity to that. And in theory, it could come back because it's probably hiding in a bird somewhere. How worried are you about the H5n1 outbreak among migratory birds in Israel? Not worried at all. These H5n1 outbreaks have occurred for years in many parts of the world. And, there are sporadically a few human cases, but it really does not become a human virus. It wipes out a lot of birds, and that's a problem, especially on farms. But, there is a vaccine for that. But I'm not worried about it in people, no. Thank you, Kevin, for your support. Really appreciate it. Is there good evidence that prior COVID infection makes subsequent reinfections, variant infections milder? Yes, there is. What happens if it doesn't? Well, if the variant changes enough to evade not only antibody, T-cell immunity, or whatever else, which I think is unlikely, then we would change the vaccine. But as I said, for influenza viruses, you only need to change a bit of the hemagglutinin, the spike, to get more serious disease. And so far, that hasn't happened with SARS-CoV-2. Now, one caveat is that the flu vaccines are not really anywhere near as good as the COVID vaccines. They're 50, 60% effectiveness against disease. So that may be part of the story. It doesn't take much to get around them. How easy or difficult would it have been for Pfizer and Moderna to tweak the final vaccine formulation during late-stage research to create efficacy drop necessitating boosters? I don't know. Are you saying that they might have done it on purpose so that they could sell boosters? I doubt it. They would have no way of knowing what they did would affect the efficacy. Are you wearing KN or 9? No, I have always worn cloth masks. And I know they're not as good as a surgical mask, but I am vaccinated. Now I'm thrice vaccinated. I'm healthy. I'm not young. And I think for me, a cloth mask is sufficient. And I've been around people who were positive. And for me, it's fine. Everyone in my neighborhood has COVID, but no hospitalization for vaxxed or not. Yeah, that's good. That's good to hear, even for the not vaccinated. That's fine. And it may be that that's a feature of Omicron, but I'm not sure we have the data to say that yet. Is there significant long-term benefit in getting a booster? I think there is, even though we don't have the long-term data yet. So what we're missing is the effect of the booster on disease, right? Well, that will come. And the question is how long lasting. And other studies have shown that we make memory B cells and T cells. And because the boost broadens the antibody response, we're going to have memory cells that are broader. So I think they will have a long-term benefit, even though we haven't done the studies yet, but obviously we have to do them. We don't know what the levels mean, but they're advocating boosters on the basis of increasing antibodies. I understand fully, Patricia, what you were trying to say. Believe me. Paul Offit said the same thing on TWIF. Please go listen to the Paul Offit episode. He said the same thing. He doesn't buy the booster for most people. He said the original 2-dose was fine protecting against severe disease. What I saw is a breadth of antibodies for Omicron. So I thought, well, if Omicron really is not neutralized by a 2-dose serum, then maybe it's a good idea to do this. And I could be wrong. Okay, I could be wrong. But I thought it was worth the shot. I understand what you're saying. But the boosters are being sold on the basis of antibodies, and I'm not sure that's correct. And Amy just said the same thing. It may be that you don't need a 3rd dose even against Omicron. We'll see. We'll see in time. And I mean, for a long time, this was my objection to the 3rd dose. I thought there weren't data to support it. I've heard 3 primers now. They use 2 to diagnose a positive with Omicron. So what was called negative before is called positive now. No, so they had a drop. So the virus genome changed, and so the PCR primers weren't working. And so they would take a negative result as a positive, right? You have a person with symptoms, and they have a dropout with that particular primer set, a lack of amplification. But that's not correct, right? Because you should know, you should get a positive result. And so then they change the primers to reflect that. I don't think they're false positives, no, not at all. Hello from Brazil. Hello, Rafael. I remember you a long time ago. I had COVID, and a year later took J&J single. 1 more dose would be enough. What brand? You could do J&J, or you could do mRNA. I think there's some possibly benefit to mixing them up. A little bit of data that suggests that's marginally good. You could, either way, it doesn't matter. Just get 1 more shot and you're set in terms of breadth of antibodies. Even though, and I know, what's her name? Patricia is going to say, but we don't know what the antibodies mean. Yeah, I know. Sometimes you have to take chances. Thank you, Neil, for your contribution. Appreciate it. Support of science education. Is there particular significance to hex in random hexamer primers? It's the right length to get. So too short would be, oh, my power is running out here. I didn't plug it in. Sorry, hold on. Sorry. My laptop was unplugged and I don't want to lose the chat on it. It's just long enough to be a good primer. If it's too short, it doesn't prime well. If it's longer, it's too specific. That's what the 6 came from. Thank you, C Service, for your contribution. Really appreciate it. Please explain why measles vaccine gives lifelong immunity from disease. So the measles vaccine, first of all, the virus doesn't change over the years. There's very little antigenic variation in measles virus. Same thing with polio virus. So the same polio vaccine has been used for 50 years. So yes, the antibody. So what happens is if you're vaccinated with measles vaccine, and let's say you encounter measles, which is pretty unlikely because there's not at least in certain parts of the world, there's not a lot of measles virus, although in other parts there are. Many countries do not vaccinate enough. Then the virus will start reproducing in your respiratory tract. That's how it enters. Actually, it reproduces in immune cells, lymphocytes in your respiratory tract, and those cross the epithelial barrier. They get into your circulation. And then your immune system detects it. You make your memory response in the form of antibodies and T cells. They attach to the virus. They neutralize it. And then the T cells will kill any virus-infected cells. So not the nose. It goes down into the lung, and gets into immune cells. And then it comes back out, and that's how you infect other people. Feeling lousy on Tuesday. Tested at home Wednesday, Thursday, Friday. All negative, then positive on Saturday. Was I contagious before my test was positive? So I think the antigen test is a good indicator of having enough virus to be potentially contagious, because not everyone is, remember. Only 20% of, 80% of infections are transmitted by 20% of people. So most people do not transmit. But putting that aside, I think you were likely transmitting at the time of your positive test, right? Maybe slightly before, because you didn't take it every hour. If the next pandemic is influenza, you think COVID will at least have prepared us? Oh, I think in many ways, yes. I think we've learned the benefit of testing early in the outbreak. I think we've learned that mRNA vaccines and even vectored vaccines can be made very quickly and tested differently than we tested before, in a way that gets the results in a good way. So yes, I think we've learned a lot. We've learned about supply chain issues. We've learned about vaccine distribution and so forth. So I'm hoping that the response will be better. No at-home tests in any store here. Sorry, my wife just ordered some from CVS. They came, she ordered it online, and they came in a couple days. So try online. Now, I told her not to get them because we're all vaccinated, and why would she want to test? But that wasn't the right thing to say to her. No. Is there a legitimate use case for using PCR for a vaccinated person rather than 2 antigen tests? Is there any credence to the recent talk of antigen tests being less effective with Omicron? So it depends on the antigen test. Some of them are okay, and some of them are affected. So the FDA has a website where they tell you which ones are working or not. They require the manufacturers to qualify them. So I think the antigen test is actually sufficient, because you need to have quite a bit of virus there, and it's most likely replicating, whereas the PCR test is going to just be positive with pieces of RNA. It's way too sensitive. So I am not a fan of PCR tests, and I think they just can detect a little bit of virus, and it's not significant in terms of transmission. So I would rather do antigen tests, and I wish we would switch to that. How's progress in vaccinating the third world? Well, it's going very slowly, right? Because there are not enough vaccine yet. It's a production issue. So we're hoping to help. Some of these new vaccines coming online are going to help that, right? This week it's been reported that neutralizing antibodies from the booster lasts less than 10 weeks. Well, that's just wrong. That's what the report said. The antibodies do not last less than 10 weeks. There's just something there that is not right, and I don't know what you're referring to. So if you got it from the news, just ignore it. The antibodies will last longer. They will not last forever. They will decline within 6 months or 8 months, but you will have memory cells, and that's more important than the actual antibodies. You will always respond to an infection with memory antibody production, no matter what your antibody levels are in your blood. Zoonosis. With respect to the zoonosis hypothesis for Omicron, would we expect on the balance of probabilities passage through an animal to decrease fitness in humans? Not necessarily, no. We would not expect that. It would be actually not unknown what would happen. And I know you're saying, well, it becomes better adapted to the animal. But it might not take much once it re-entered a human to regain that fitness, because the viruses make a lot of changes. So I don't think you could say one way or the other. Thank you, Stephanie, for your contribution. Amy's a rock star. Yes, absolutely. First saw me on the David Pakman show. Yeah, I got invited back on the Pakman show. So we're going to do one of those. They want to talk about, Omicron. Okay, we'll talk about it. Yes, good questions, I think. No, don't worry about the bird flu outbreak. It's not an issue. Not going to happen. Please comment on the Army COVID vaccine. So this is the ferritin nanoparticle. They fused the receptor binding domain of spike, only a small part of spike, to ferritin protein. It makes nanoparticles, multivalent, lots of copies of the RBD. And it works well at inducing antibodies at T cells that react with many variants. That's great. I think we need more vaccines. I don't think it's any better than what we have, but it's another vaccine that works. That's my feeling. How is the PCR validated to diagnose disease? Well, you have the virus that you've recently isolated, SARS-CoV-2, in the lab. You design the PCR test, and you calibrate it with different amounts of virus, RNA, to make sure it's giving you the sensitivity that you need and the copy number detection that you need. We have had a lot of stupidity in this pandemic. Amen. Absolutely. My favorite today, where Bill Hazeltine said we overemphasize these vaccines and didn't emphasize monoclonals and N95s enough. This is pure, utter BS. Dr. Hazeltine. It's nonsense. Vaccines are the way out, not monoclonals. Monoclonals are for people who either can't get vaccines or don't want them or decided or get sick even though they are vaccinated, and then you could save their lives. And the N95s, you should use them anyway, for sure, but not in place of vaccines. No. So Henrik says, have you read the preprint? And I have not, but his conclusion is the rapid spread can primarily be ascribed to immune evasiveness rather than an inherent increase in basic transmissibility. What did I just say 10 minutes ago, folks? Thank you. I would buy the immune evasiveness, absolutely. But don't remember there's a human factor as well. That's good. I'm going to read that. Let's see here. I'm going to try and move through these to get, because I'm not very far in. That's not good. Small stockpile, smallpox. Any other vaccine stockpiled? No. That's the only one because the virus has been eradicated and we're afraid it might be reintroduced by bioterrorism. How concerned should I be to fly cross-country to see grandchildren all under 3 and unvaccinated if I'm vaccinated and boosted? Can I get virus on the plane and infect the grandkids in the first 5 days? So if you are concerned, you could take some rapid tests, right? Take one when you get there, wear a mask, and if it's like twice negative, then you're good to go. That would be what I would do. Can you comment on small molecules targeting things besides the polymerase and proteases? I'm not aware of any others that have progressed as far as that. I think a frame shift inhibitor is not going to happen anytime soon. The endonuclease, the correction enzyme, is a good candidate. And that's about it. Okay, Pfizer on 4/1 and 4/26, so 3 weeks. Booster on 12/30. 2 months late. It's good. Later is not a problem. You're good to go. Yeah, Daniel says never miss a chance to test, but if you listen to our conversations, he agrees that we are testing in certain cases too much. With low affinity antibodies, are antibodies less likely to bind as they just drift by? No, they detach easily, so the off rate is higher than the on rate, yeah. I don't know. Maybe he did say never miss a chance to vaccinate. Yeah, but he's circumspect about testing. Listen to the last one. He and I get in conversations where he recognizes that we're doing too much testing. We test to protect 94 and 98-year-old. You could do that if you have them in your home, yes. But there's also this idea that we're testing to protect the unvaccinated, and I don't agree with that. I think they should be vaccinated. You may differ in your thoughts, but that's how I feel about it. Joan says Omicron's so infectious, almost all will be exposed, and that's not correct to say it's so infectious. So smaller loads are is not known to do anything different. None of this is correct to say it's more infectious. As you just saw, it's actually the antibody evasion that is responsible for its drive through the population. These are the things that I deal with on a daily basis. I'm trying to counter, but it's very hard to do because you hear this over and over from other people and very less frequently from me. I am 3 times vaccinated, started symptoms Monday. My antigen test is negative. You have asthma. I would say, since you have symptoms, just try an antigen test every other day to see when you're going to be negative so you're not a threat to other people. But don't hide in bed. You don't need to stay in bed, but I would say stay home so you're not going to stress yourself since you do have asthma. How would you advise the sports league how they should proceed for the next 2 months since they have to test all the time and stay negative? Well, it would be good if you could put them in a bubble, right? If you really want to play, keep them away from everyone else and test them frequently. I think if you want to keep them negative. On the other hand, if they are 3 times vaccinated and they're infected but asymptomatic, I don't know what the difference is on the team itself. So at some point, we have to start changing these ideas that you have to test everyone and we have vaccines, folks. We have 3 times vaccines. Most people are going to be fine, especially athletes. So I would advise them to relax it a little bit. If I have this correct, the only reason we need a booster is because they gave the second dose too quickly. That is my opinion. That's what we concluded on TWIV. Now, the CDC will say, no, it's to keep you from getting infected, to keep your antibody levels high. That's baloney because your antibodies are going to go down in 6 months. So what are you going to do, get a booster every 6 months? So yes, the first 2 were mistimed. We're fixing it. That's how I view it. And Amy would agree. Nothing on Omicron and long COVID. No. But as Daniel said, the vaccine infections of vaccinated people, much, much lower rates of long COVID. Do virus mutations over time cause them to lose virulence? I haven't seen that happening. I think that's the theory that it would happen to increase transmissibility, but I haven't seen that. All right, we have a double vax family, mild COVID end of November. So you're good because you have both recovered and COVID, so I think you have the broad response. It depends where you want to go, if there's a lot of COVID or not. And if you're going to have to, and it depends on their regulations as well. I wouldn't necessarily cancel it, no. I mean, I really do think that with the vaccines we have, you can start doing normal things. I've been to restaurants. I've been to theater. I think if you do it properly, it's fine. How does protection from multiple infections compare to spaced vaccines? My son was infected in June, then 3 weeks ago. That's a good question. I don't know. The only real good data we have is on recovery and then vaccinated, or vaccinated and then infected. Actually, so that's what your son was not vaccinated. So here's the problem with recovery from infection. Immunity after recovery is good, but it's highly variable depending on the person. And so that's why we suspect that a vaccine dose is a good thing to get. Thank you. You don't have to have a question. You can listen, and I appreciate the thanks. What is a recitation? Well, a recitation is typically done in a course when you have the lectures given by the professor, and then a teaching assistant will take groups of students separately, and they'll read and discuss papers. And the TA will walk the students through the papers and answer questions and talk about methods and so forth. So it's a little hands-on way to get more information about how the information that the professor told you was gotten. What's the difference between a non-sterilizing and a leaky vaccine? I think that's the same thing. Non-sterilizing and leaky. Leaky is a crappy word for it, but non-sterilizing is the right one. My 4-year-old came in contact with saliva from a utensil of a fully vaccinated family member who tested positive. Is virus in the saliva? How long should we be concerned? Well, I don't know what kind of contact. Did it just touch their hands? Did it get into their nose? Or maybe you don't know. Fully vaccinated person is at best shedding for a day or two, I would think, and we don't even know if it's that much virus. So I don't think you should be concerned. Just test. Give them a rapid test if you're worried, because if you're concerned about illness and they tested positive, you could consider monoclonals maybe, or maybe not approved in that age group. So no, cancel that. Just be careful. I think you'll be fine. You should be all vaccinated to begin with. I think so. We have vaccines, and there's really no reason to have 30% of the U.S. unvaccinated or whatever it is. But it's not up to me, right? Saying that is not realistic. I understand that. But I'm not going to be careful because of unvaccinated people. I don't want to hurt people, but I think it's unfair in many ways. NBC is stating the home rapid tests are not able to pick up. That's BS of the highest order. Some of them do. The ones that don't can't be sold anymore. Isn't the E at the end of panettone supposed to be voiced? No, it's panettone. It's not panettone. It's panettone. Okay? That's how I was raised, by people who were Italian. My father was born in Italy. I am first generation, and so was my mother. How does AstraZeneca compare? It's nearly as good. So I don't think people are screwed. No. Is original antigenic sin happening with Omicron? No, it's not. Original antigenic sin is when, say, you get vaccinated, then you get infected with a variant, and then you make an immune response to the original vaccine. Well, that's normal, right? The problem occurs with influenza, when the virus you get infected with is very different from the original vaccine, and that doesn't do you any good. But with SARS-CoV-2, the response is good. It's not a problem. At a large event like a convention where everyone is required to be vaccinated, does it make sense to require masks? The vaccines aren't 100%. So if there's a high level of virus there, and it really depends on what's circulating at the time. There are times when circulation levels are low. But if it's high in the community, or wherever the people have come from, it makes sense, because the 10% of the people could get severe disease and die. So you should, in theory, protect them. That's the one reason why I would think it makes sense. Or if you have young kids at home and you're going to be home in a few days and you worry about them, it makes sense to wear a mask as well. But for you should be fine, except for that 10% or so, right? Yes, the talk of pastries. Your talk, whatever it takes to get people vaxxed philosophy, hindered my embrace. Well, I'm sorry. I am a proponent of vaccines, and I think you should get vaccinated unless you have a good reason. If you have a medical reason not to, that's fine. But why not? Don't tell me it's my right to decide. That's BS. You can't have a right to overburden hospitals and stress out medical staff. It's just ridiculous argument. And I don't care that it hindered your embrace. I'm sorry. But this is the way this pandemic ends, and it could be over now if people all mostly got vaccinated, not all even. Should we open up society? I think it's opening up, and that's why we have more infections now, because we are opening up more than we were. Understand that. Can exposure to a small viral load create antibodies without causing illness? No, I don't believe so. I think exposure to a small viral load, if it gets cleared by your innate system, you're not going to have any adaptive immunity. And if it takes hold enough to give you adaptive immunity, you're going to get the virus is going to reproduce in you. So I don't think a low load has ever been shown to be correlative with disease and with immune responses. Coronavirus can spread to months. So this is not correct to say it can survive for months because they're looking at viral RNA, and you have no idea if that's infectious virus. It's just pieces of RNA, which happens with many RNA virus infections. Yeah, you can see viral RNA in many organs a long time after the infection is resolved. What does that mean? Probably nothing. So be careful with PCR. My daughter is quarantined for 10 days, testing positive with no symptoms after being back twice. 10 days is really the limit of antigen positivity. I'm surprised. I think should be negative. So I think you're at the outer limit of being antigen positive. So I think it's unlikely. It's the only channel in the world that covers COVID and panettone. Yeah. Well, we're human, right? Do you see new variants causing highly pathogenic strains that can complete? No, absolutely not. No, not at all. And so far, we've had no signal of that, and there's no reason to think it's going to happen. It's never happened with any other virus. So I base my prediction on all of that. I could be wrong, but I think it's unlikely. I have to convince my nephew to get into micro. Yeah, expose your nephew to all this good stuff. Not viruses, but information about viruses. My daughter tested positive, vaccinated twice. She quarantined for 10 days. I think vaccinated twice, 10-day quarantine. No, not transmitting anymore. Why are you saying stop testing when folks that are vaccinated are still catching it? No, you're not a spreader. For most likely, most people vaccinated do not spread. A small fraction of them do, and at most for 1 or 2 days. And most of them are asymptomatic. What's the point? Do you test for flu every year? No. You get flu, you get a sniffle. If you get serious flu, you go to the hospital and get tested. Most of the time, you don't get tested. And if we just randomly tested people for flu every year, it would be a nightmare. So that's my argument. I want this to be almost a cold virus now in most people who are vaccinated. There's no reason to test for it. Unless you want to protect unvaccinated people. If that's your goal, that's fine. It's not my decision. In theory versus hypothetically, yeah. People don't listen to me when I correct them, but I want to use proper terminology because I think it's important in scientific communication to be precise. And, people tell me, English is a fluid language. That's fine. Science is not that way. You have to use precise terms. And when I write a textbook, the word I say in chapter 1 has to mean the same thing in the last chapter. Otherwise, there's no sense, right? Will one dose help against Omicron for severe disease if it's recent? So it may not neutralize Omicron, right? Because you need the maturing of the antibody response, which comes after 2 spaced doses, or if not spaced correctly, 3 correctly spaced. So I think it's not a given that you're going to be protected after just 1 dose. Walk up testing sites with people who are likely exposed or not feeling well. What could go wrong? Well, then they test positive and their employer says you got to stay home, or they can't travel to see people they want to travel. They close universities, right, when there's a lot of kids who are testing positive, but none of them are symptomatic. What sense does that make? They send them home to bring whatever virus they have with them. I think this is not a good strategy, and Daniel Griffin agreed with me. I'm not making this up. He said we're testing too many people. What's the definition of mutation? Is a change in nucleotide sequence, DNA or RNA, not protein. I'm sorry, you're all wrong who say the mutation in the spike protein. It's not right. Mutation was originally defined to mean a change in DNA or RNA because it was recognized as that, and there's no need to expand it. An amino acid change is just that. It's not a mutation. Is age 30 too late for HPV? I think the recommended age is 26. Yeah, I don't. It's probably less useful at that age, right? But I don't think you cannot have it in this case, with one partner. So, what would happen if all the viruses on Earth were deactivated at once? I would say in about a month, life would cease. I love viruses, so I could be wrong. Certainly bacteria, if you took them all away, life would cease, right? How long should after COVID recovery, should you get a shingles vaccine? So Daniel just answered this recently. I think he said that there's no need to wait till the shingles is over. Just get your vaccine when you can. There's no contraindication. Thank you for your support, DR. Is canine rabies vaccine sterilizing? If not, how much replication can happen before irreversible disease? I doubt that it is. I don't actually know for sure. But the point is that, the incubation period for rabies is long, and it takes a long time for the virus to get into the CNS. So even if a vaccine is not sterilizing, as long as it keeps viral loads low and then eventually eliminates it, it won't have enough time to get into the CNS and cause it issues. 2 Coronavac shots and a Pfizer. So, we don't have a lot of data on that combination. But if the Pfizer was given at least 6 months after the Coronavac, it should be okay. In the UK, we have the worst virus, the Boris Johnson virus. If deer can contract COVID, does that possibly extend to reindeer? Possibly, yes. Are they closely related? Let's see. Are deer related to reindeer? Reindeer and caribou are the same animal and are a member of the deer family. Yeah, in North America, they're called caribou. They're wild reindeer. They're quite close, members of the same family. And, with viruses, if you're in the same family, you're quite close. So it's possible. I think people need to look at that. Yeah. You're asking me about this transmission in Danish households. I haven't read it. I will check it out before the next live stream. I'm halfway through these questions. Why does a new variant displace an older one? If it's more infectious instead of both variants being present or even one person. So variant displaces an older one. We know this very well for influenza virus because every year variants arise that displace previous ones. And they're never called more infectious or more transmissible. They're antigenically different. So they evade immunity slightly, and that's enough to make them fit. Why SARS-CoV-2 variants displace others, why one is more fit than another, is not known. It could be antigenic character, and that's what we just mentioned briefly before in one of those preprint comments. But it could be something else. It could be how much virus is produced, how fast the reproduction cycle is. For Delta, it was thought that it reproduces really quickly, right? And so you're likely to shed before you even have symptoms. So that would infect people more efficiently. It's not inherently more transmissible, right? But in the lab, that wasn't held up in cells from humans in culture. The Delta replicated just the way the other variants did. So we don't know why it displaced it. Understanding that is hard. So it can be multiple properties of the virus that would explain it displacing another. And what does displacing mean? Well, if you put two viruses together in a cell culture in a lab, one wins. And so in a population, one virus would win at a population level. What does that mean? Well, that's why we have to figure out what fitness means. I had Regeneron treatment last month. It's at 90 days. Yeah, that's what still the number is 90. Yep. How do you get 30 mutations without a missense? Well, the missenses happened, but those were weeded out, right? Those viruses never made it. So that's how you have to look at it. Well, the bad mutations, the negative ones, they're gone. Those viruses are dead in the water. Chris, thank you for your contribution. I had one shot in April, shot 2 in July, currently have COVID. Should I still get my booster? I don't think you need a booster. You've got a COVID booster. 2 shots in the COVID is good, in my opinion. Okay, let's see. The studies showing Omicron isn't replicating in lung tissue very well. So first of all, Eric Topol is a cardiologist, doesn't know what he's talking about. Okay, and I don't want to be mean. Stay in your lane, okay? He does not know how to interpret virological data. And he's got 250,000 Twitter followers, and this is a crime because people think he's preaching the truth. And the 5 studies showing this are all wrong. They're not done properly. And that Hong Kong study is wrong. And none of them are peer-reviewed, and I bet they don't make it through peer review. So please be suspicious of early data that hasn't been looked at. For example, there's no time zero. This is a flaw in the experiments. You can't conclude anything without a time zero. They have to report PFU per cell, not PFU per milliliter. That is meaningless. And after that, if you still find it replicates poorly, then I'll believe it. But I've been doing virology for 40 years, and those experiments are wrong. I am sick with COVID-like stuff, feel bad, negative antigen test, can't get a PCR. 3rd vaccinated, I have asthma. Should I keep trying to get a test? Well, you have to ask yourself, what would that do for you? Because you're triple vaccinated. Are you worried about infecting someone else? Then you should get tested. You're going to recover, probably in 5 days or so. Maybe a little longer because you have asthma. Take it easy. Get rest, eat well, stay away from alcohol. And if not necessary, you don't need to be tested. Now, if you get worse, if you can't breathe, you're going to go to a hospital. They're going to test you for that at that point. But this is a case where it seems to me that testing doesn't provide useful information. It's just maybe psychological. Oh, I have COVID. Now I feel better. I don't get that. No. As it seems, vaccines don't protect well against infection, which is fine. It protects against disease. Wouldn't everyone get infected in the end? Eventually. But, I think the shedding in a vaccinated person is lower. The likelihood of transmission is lower. So it might take a long time. And as you bring down virus loads in the population by having a lot of people vaccinated, then it gets possible that there are pockets of people that never see the virus. Yeah. But right now it's not happening because there's so much virus around. Is it possible that antibodies against HPV stay because of frequent re-exposure? High levels, you mean? I don't think there's any study that shows that the sterilizing effect has to do with that, which would correlate right with sexual activity. I just think that the antigen is really different, and for some reason that maintains high levels. Yeah, but I mean, it's a good idea. And certainly re-exposure would do it, but I don't think everyone who has high levels gets re-exposed. Do you think it would be better messaging to use the word resistance instead of immunity? Resistance also implies no infection, right? But I do think this is a messaging problem because most people think vaccines stop infection. And the CDC has made it worse by saying we need the boost to stop infection, right? it does seem that Omicron is more transmissive and fit. No, it's more fit. So Bob, why do you say it does seem? I've told you reasons why it could not be that. It might be something else. And yet you insist on saying it does seem. This is what I have a problem with many people who I talk with this about. There are alternative explanations. It is more fit, and why it is we don't know. And it's probably antigenic evasion, as you saw in the preprint. So I don't understand why you have to accept this when there are alternative explanations. Since virus has DNA or RNA, why are we not getting those code to find a way to defeat them? Oh, no, we are. That's how we made the vaccines. We got the genome sequence of SARS-CoV-2, and we made vaccines with that. So, yes. My 7-year-old's getting dose 1 tomorrow. Should I delay second dose? She had a mild case of COVID a year ago. So they're not going to want to delay it. The recommendation is 3 to 4 weeks, right? You're going to have a hard time delaying it. Since you had an infection, I think it would be fine. It would be safe to delay it for up to 6 months, and then you get that broadened antibody response. So I think it would be okay given the fact that you've had an infection in 1 dose. Does anyone teach virology to students younger than high school? No, not in a class. No, not much. Probably mentioning it, but it's usually wrong. I mean, even high school, it's hard to find. Even college. Not every college has a virology course. Columbia University did not have a virology course 12 years ago when I started teaching my virology course. Can you believe it? I tell my students their parents pay $60,000 a year in tuition, and they didn't have a virology course? Come on. And when I leave, they probably won't have one either. So, I mean, now I teach my course online so the world can take it. What are you waiting for? Don't tell me you don't have time. I have time to do like 6 hours of teaching a day some days. It's because I love it, I know. In terms of infectivity, is there a difference between an unvaccinated and a vaccinated with a negative rapid test? Well, is it an antigen test? Then I think there's no infectivity there with the antigen test. If it were a PCR test, it could also not be infectious, depending on how much RNA is detected by the PCR test. But I do think a vaccinated person is shedding less virus for a shorter period of time after they're exposed to virus. How long after the booster is the vaccine effective? Yeah, 14 days. I mean, look, 10 to 14 is fine. It really depends. It's not a fixed number. It depends on the person. But, in the vaccine trials, after 11 days, you started to see a difference between the vaccine and the placebo groups. So certainly 10 to 14 days is fair. Would different variants have different surface transmission characteristics? Well, in theory it could, but I don't think that the transmission mode has changed. I think it's still in droplets, respiratory droplets. What is this about? The mRNA vaccine reprograms adaptive and innate responses. Yeah, I saw this paper a long time ago. They're just using weird... They think it's reprogramming innate responses. I think the data they have for it doesn't support that whatsoever. I just don't think it's correct. A lot of the stuff that comes out is not necessarily right. I hope you realize that. And I'm not just criticizing it because I'm a crank or grumpy. No, I'm trying to get you to see that science is not always on point, and especially in a pandemic, right? The Binax now at homes have a short expiration date. So that's because that's as long as they've tested them. They put them on the shelf for however months, and they test, and it's okay. And they say, okay, that's the limit. And it's a lot with like other expirations, but you can use them beyond that. I use milk months beyond the expiration date. It's still fine. Well, then eventually it goes off. But there are other things that are fine for a long time. It's only because they haven't been tested for that length of time that they can't qualify it for that. What's the difference between serotype and variant? So a variant is any virus that differs from another one. So if I have COVID and they isolate virus from me, and then you get virus from you, and they sequence both of them, and there's one nucleotide difference, that's a variant because it's one nucleotide different is enough. There could be 10, there could be 30, 100, doesn't matter. A serotype are two viruses that differ in their reactivity, say, with antibodies. So if I take antibodies from a vaccinated person and I react them with one virus and it doesn't react, and with the other it does, they could be different serotypes because they have different reactivity. So but there are 3 serotypes of poliovirus. So if you get infected with polio type 1 and recover, you're not protected against types 2 or 3. And that's why the vaccine has to have all 3 serotypes in it. Here's a question I saw before. How is the average person supposed to know whether Malone is on the level or not? You can't know. And not even the press can know, right? You hear this guy and you say, oh, okay. So what do? You look for scientists who talk about it, like us. That's the void I'm trying to fill. I'm trying to provide some kind of science education that would get around this stuff. And it can't be just me. That's why I started an incubator, and I'm trying to raise money to hire people to help me. I think there's a future for that. Because you're right. The average person has no idea when people get on and say the virus came from a lab. And they say, oh, this guy's a professor somewhere. This lady's a professor. How are you supposed to know? That's an issue. Hello, Ian. I am on a B-cell depleting therapy for MS, yes. In New Zealand, I had a third primary Pfizer. To time it for infusion, it was 8 weeks after the second dose. So 2 months is not optimal. It's better than a month. 6 months is the best, right, for that third dose. So I would say you have not the optimal antibody response with respect to neutralizing viruses. However, as we have talked about tonight a lot, it may not matter. In terms of disease, you might still be protected. And we don't have the data on that yet. Certainly not with Omicron. So again, Steve, just throw away the Twitter, okay? Monica Gandhi, respect for Monica Gandhi, not just because of increasing immunity, seems less virulent, is totally wrong to say that. You cannot say it's less virulent from the limited studies that we've done so far. In the end, folks, none of this is going to matter. Just get vaccinated and this will end. There's always a reason. So MI says, in Israel, the holidays were over some time ago. There's always a reason you don't know about. People are getting together, whatever. And you say, oh, it must be the virus. That's not scientific MI. I'm sorry. Boy, you guys are pushing me to crankiness tonight. Not really. Do you think you would go on Dr. Peterson's podcast if invited to talk about COVID? Well, sure. I will go on reasonable podcasts to talk about COVID. I don't even need to know the questions ahead of time. I will just tell them what I tell you. Why the deer? Like, how do you think they'd be sufficiently socially distanced from humans? They're not, actually. And listen to the next TWIV. You'll see we talked about this quite a bit. They're very close to humans, actually, in many ways. Yep. And Tom answers that, but we have a whole half of a TWIV on it coming out. Yeah. A new paper says Omicron is still susceptible to our T cells. Made me think of you guys when everyone was freaking about it. Yes, exactly right. This is what we said from the first day of Omicron. The T cells will save you. They're going to save you. We're usually right, folks. But you don't have to believe us. You can just wait for the data to come out. Do second and third doses have any effect on somatic hypermutation? They do. They allow it to proceed uninterrupted. Yes. And that's why spacing is important, to allow somatic hypermutation to occur. You would pay money to see me and Amy playing whack-a-mole. Well, it's fun. I like it. My carer just told me she'd been quite badly ill with COVID. Same house her vaccinated husband didn't catch it, nor her unvaccinated son. Well, as I said, not everyone transmits. 80% of transmissions are by 20% of infected people. So just take that, and every time you hear these stories, that explains it. Not everyone transmits by far. Does the current vaccine also protect against SARS-1 and MERS? No. Maybe to some extent from T-cell reactivity, but not enough. No, it wouldn't be sufficient. What does it mean when Putin says Omicron is a vaccine? Now Putin has become a virologist. He's following the mantra, the narrative that many of you are listening to on the news. This is not virulent. It's highly transmissible, so everyone's going to get it and they're going to be vaccinated. That's what he's talking about. It's all BS. It's BS. Why? In part because, well, we've talked about it. Yeah, you infect vaccinated people, that's mild. That's the way it works. That's the way vaccines work. I don't think there are benefits of a DNA vaccine over an mRNA vaccine, frankly. I think the mRNA vaccines they're quicker to make protein. They make more protein. DNA vaccines were tried for many years. They didn't work well. So for humans, I think they're done. Your views have not changed with respect to immunity. Have you reflected on this? What do you want me to reflect? I think that scientifically was correct to the start, and it's correct now. And the data have been consistent with what we think. Not that we're brilliant. It's just that we're looking at the data. We have been virologists all our lives. You understand? We think about viruses. I have thought about them since 1979. No, before that, 1975. It's a lot of years, every day thinking about them. I'm not a cardiologist that's a year ago got interested in viruses and who never thought about them before. It's my field. I know what I'm talking about. I'm not always right, but I get it. Should countries just stop testing en masse? No, I think there are cases where you have to test. If you have an unvaccinated family and someone is sick, you got to test them to see if you should separate them from everyone else. If you have a patient in a hospital, you have to test them because there are unvaccinated people in hospital. There's situations where you have to test. But you have a university, what the hell? If all the kids are vaccinated and they're required to be vaccinated to show up, what the hell are you testing them every week for? And then they're positive with no symptoms and you send them home early so they don't get stuck from quarantine. It's all ridiculous. But no, not blanket no testing. Just think more carefully about it. Thanks for your contribution. If we get negative antigen tests and are symptomatic, should we assume it could be Omicron? It depends on the test. You have to look at the FDA site and see what tests have been declared to not work. You could have other virus infections, that cause similar symptoms. It's winter. That's what viruses do. Here in the U.S., in the temperate climates, they cause infections during the winter. Okay, thank you, and Prisco, for your contribution. You said it's possible to get COVID after harvesting a deer. What about surgeons during surgery? That's why they wear face masks. You'll find a surgeon not going to take their face mask off to protect you, but they're also being protected. But if you have COVID, they're not likely to be doing surgery on you, right? I know this a bit because my dad was a surgeon. Before you jump all over me. Now we're just at the freedom of information. I think Questella puts it. Surgeons are wearing more protective stuff. People rip open deer barehanded. Well, I guess that's true, right? That's so funny. I believe the notion of viruses tending to vary into less virulent, less dangerous is a myth. I think it is an idea that makes sense to us humans, but there is no evidence to support it. There's one story, which is the introduction of myxomavirus into Australia to kill excess numbers of rabbits. In the first year, 99.9% of the rabbits were killed when the virus was released. After that, the virus and the rabbits changed to be more resistant. And the virus became less virulent and the rabbits more resistant. So that could happen, but we've never seen it with humans. Two people are in a room, one vaccinated, the other not, and neither has any symptoms. Who is more at risk for what? At risk for what? Getting infected. They're both at risk for getting infected. I don't know who's going to bring the virus in. Who's at risk for severe disease, hospitalization, and death? The unvaccinated person. It's always the case, folks. Most of these cases you see, they're in unvaccinated people. Go look at the New York Times curve. It tells you that. They take data from 26 states. Most of the cases are in unvaccinated. Most of the hospitalizations are in unvaccinated. You know, we can talk about this till the crows come home. Or no, that's cows. But the fact is, vaccination stops all of it. We wouldn't be talking about Omicron if more people were vaccinated. Thank you for your contribution, Daniel. Books on the history of virology or particular viruses. Let's see what I have on my shelf here. By the way, speaking of books, so I have a nice collection of virology books here. All right, so this is by my colleague Ann Skalka. It's all about the history of retroviruses. It's very good. She's a good writer. She co-wrote the textbook of virology with me. And this one, we're talking about evolution of virulence. This guy, Paul Ewald, has written a book about this, several things actually. Evolution of infectious disease. He talks all about the theory. You should read The Cutter Incident. It's about failure of the first polio vaccine. All right, last one. Fever. Get it. It's awesome. Made me want to be a virologist. Is it possible that the plant viruses I eat protect me from other infections? It's possible, but I don't see any evidence for it. In fact, you know what the most abundant human fecal virus is? Pepper mild mottle virus. Yeah, because we all eat a lot of peppers. It's possible, but I don't see I don't see any evidence for it. So Mario can't remember any of his questions. Write them down. Write them down. Okay, New Zealand is the first to get the new year. Everyone's now saying this like an hour after. Christmas Island, the Marianas. Cool. All right, let's buzz through here and try and get back to the end again because I don't want to keep you forever. Almost 1,000 people. Wow. That's cool. R0 only applies to trials. No, R0 is calculated early in an outbreak. It was first calculated in the very first month of the COVID pandemic in China. 2 to 3. And every subsequent recalculation, which is RE or RT, doesn't take into account the human behavior that varies at different times. So you can't say that this is all virus changes that is causing this fitness. And as I said before, you can reduce the R0. So how can it be much of an indicator of the intrinsic transmissibility of virus, which intrinsic means you can't change it. It's an inherent property of the virus. Someone in news article comment asserted scientists wouldn't say about he'd be appalled at squishy. Squishy is a great word. R0 is squishy. How come we can't make a vaccine for AIDS? Okay, short story is vaccines are not sterilizing. The problem is when you get infected with HIV, it's not sterilizing if you've been vaccinated. And so then a fraction of the viruses are going to integrate into your cells, and that's it. You're done. So it has to be sterilizing, and they haven't been because they're just it's hard to make a sterilizing vaccine. And the only one is the HPV vaccine. Thank you, Stephanie, for your contribution. Appreciate it. Thank you, Marge716, for your contribution, for your support. Really appreciate it. What should the CDC do about cruise ships? Well, I think it's up to the cruise ship companies. They're not going to want to make vaccination required, right? Because then they'll lose business. So they want to have a mixture of people. You're talking about with respect to COVID. I don't have a problem with it because if you're vaccinated, you could go on a cruise and you're 90% chance you're going to be fine. So I do think we're at the point where we need to get going here, and we're not going to have unvaccinated people slow us down. How does our body know to produce IgA after natural infection? Well, there are two kinds of IgA. There's IgA that's circulating in your blood, so they could be made by B cells anywhere. The B cells are instructed in the lymph nodes, and then they move out and produce antibodies in the circulation and in tissues. And then there are lymph nodes near mucosal surfaces, like under your throat. They service the nasopharyngeal mucosa, and they will make IgA-producing B cells that are secreted as well. So it's really made in a lot of places, and then certain kinds can get targeted to be secreted across the membrane. Yeah, Amy has to leave after an hour. We have done some ones where she stays to the end, but usually that's when we're in New York, and I rarely do that. Polio survived treated swimming pools. Yeah, soap. We put high concentrations of soap in with polio, and it completely does not lose its infectivity. Thank you, Sylvia, for your contribution. Really appreciate it. I don't, I'm sorry if you're confused about testing, but if you were vaccinated, especially triple, you're not likely to get very sick, and you should be able to, you don't need to be tested. I don't see the point of that. But there are other cases where you do want to be tested. The Metropolitan Opera is requiring all cast and crew to have 3 doses. That's interesting, 3. So I went to a musical a couple of weeks ago, and 2 were sufficient. Restaurants, 2. Well, that's fine. I don't have a problem with that. Okay, let's see here. Amy has just left. There are a lot of great questions here, but I think we need to wrap it up. All right, let's see. Many years ago, an infection control nurse emphasized that friction was the key. I think this is important for bacterial contamination, but viruses, it's going to be harder to get them off. Not impossible, but it is harder. Someone said, stop talking and play the guitar. I think I'm better at talking than playing the guitar, right? If I went to a restaurant and saw the cook sneezing and coughing and ate the food, could I get COVID? Well, it depended on whether the food was then cooked if it was heated after the sneezing and coughing or not. But when you eat, you aerosolize, right? And you could potentially be infected if there's enough virus in there. But remember, everyone doesn't transmit. Nope. Back to the nucleic capsid. If the immune system generates antibodies against it, I'll stop at 10:30, folks. I'm sorry to keep the mods here, but if you want to go, that's fine. How would the antibodies reach the nucleic capsid since they're either in the cytosol or inside the virus? Well, they don't, right? And that's why antibodies against nucleic capsid are probably not blocking virus infection. Because, yeah, unless they could do something inside the cell, which is possible. Some antibodies work inside the cell. Well, we don't know if that would happen for nucleic capsid. It's possible, but we just don't know. But that's a good point. Very good point. Thank you, C Service, for your contribution. You helped me overcome vaccine hesitancy. How will 2 recent Modernas hold up to Omicron compared to vaccinated in spring with recent booster? So we've been talking about this all night. 2 closely spaced Moderna shots. The antibodies you make don't neutralize Omicron very well. They take a hit, whereas the 3rd dose broadens the response to include Omicron, which has changes in antibody epitopes. However, what does that mean for disease? We don't know yet. It's going to take some weeks and months to get the data in. So it can't say that you're fine with 2 against them. You might be. There are some people who believe that it doesn't matter what the neutralizing titer is. And that's heresy in many cases, because the CDC and Dr. Fauci are all saying you have to have boosters that get your antibodies up. It may be that that's not the case. So I think you need to stay tuned. Can you debunk this particular notion that mRNA shots, the spike generated by lymph node cells, makes lymphocytes kill them and leaves our immune system in a wreck? Well, no, it doesn't leave our immune system in a wreck. I mean, in fact, yeah, some cells that produce the spike may be killed, but it's going to be a small number of cells in your muscle cells mainly. The other cells, the immune cells that present the spike, do so in a degraded form. The spike is chopped up. It's not the whole spike, and it's presented to immune cells that way. So there's no way that the majority of your immune system is going to get trashed as a consequence. I mean, the immune system is always presenting antigens, presenting proteins to make an immune response against them. And it's the same as with any other protein, and they're not trashed, they're not killed. So that cannot happen. Thank you, Overjoyed, for your contribution. If getting boosted every 6 months would prevent infection, I'm on board. You could do that if you wanted, and I understand. I mean, we talked about this a little bit with Daniel, and he thinks that the rate of LH, long haul COVID, is so low after vaccination that it's not an issue. I think we need more data to sort that out. It's a really good point, though. I agree. With 1 shot of Moderna, should I wait 6 months? All right, so the problem is if you wait 6 months, you could get infected and you might have more severe disease because 1 dose doesn't give you very good protection, right? So if you have the ability to get 2 doses and a booster 6 months later, that's perfect, with all the caveats we've said tonight, right? It's definitely behavior in Arizona. Thank you, Margot. Little things make a difference, and it's human in the end. Restaurants are packed probably more than prior. Yeah, people want to get back to their life, and that's what's driving this outbreak. It's not the bloody virus, folks, and many people get it. Behavior. And you know what? If the virus is shown to be somehow more transmissible, I'd agree, but I haven't seen it, and I haven't seen it for any of the other variants. And don't tell me that population observation proves it, because it doesn't. And Ron Fouché, an expert on viral transmission, agrees. Thank you, Faster Tortoise. Good night, and thank you for your support. Can COVID be transmitted to someone by sharing a food utensil? I think it's low probability, even though you could aerosol it. Our breathing and eating tubes are very close together, so the virus could get up there. I think it's low probability. Sandy, thank you for your support. Really appreciate it. Thank you, Sylvia, for your support. I'm confused about the stop testing. Is it not a good to test before attending a gathering? So if you're triple vaccinated, what are you worried about? Are you worried about the 10% probability that you might have severe disease? If so, then you should mask up before going to the event. What's the testing going to do? Everyone's vaccinated at the event. What is the testing? It's like a school of kids who are all required to be vaccinated. Why are you testing them every week? We don't test for other infectious diseases routinely during the season. Why are we doing it now? This is now a vaccine-preventable infection. That's why you don't have to test everyone. Was frequent, widely available testing not a good way to prevent putting life on hold? Yeah, before we had a vaccine. We have a vaccine now. That's the game changer. The vaccine does it. Heterologous prime boost, if it looks like ad vector followed by mRNA may lead to some enhanced immunity. I think the reverse would be true as well. And then maybe it's the T cell boosting. Yeah. I'm just not sure. I'm not going to answer that one. My aunt was refused monoclonals because the doctor said they don't work against Omicron. Well, of course, if they didn't test, that's absurd. But some of the monoclonals don't. But some are clearly known to, including the GlaxoSmithKline one that lasts a long time. But there, in the latest TWIV report, Daniel Griffin clearly said that he gave a link to a chart which showed which work and which don't, and clinicians should be aware of that for sure. Yeah, it's not really appropriate not to be. No Agenda used the soundbite of Daniels to misrepresent facts. Yeah, well, No Agenda is trash. I've known that show for many years. They don't know what they're talking about. Thank you, William. Glad you're learning, and thanks for your support. Yeah, No Agenda is Adam, what's his name? Yeah, I mean, they should know better, but they have a history of trashing vaccines, and so do other tech podcasters as well. And by the way, folks, we are not having a deluge of poor information being spread. This is the best place where you can get information about viruses. Does EBV remain latent in the body? Yes. It remains latent actually in memory B cells. And so is it driving CFS? It could. It could be, but I think it's more complicated than that. And I think we're going to see some interesting information about that in the next year or so. Nathan, thank you for your support. Would I debate this guy on JRE? No, I am not debating anyone who is spreading misinformation because it only it dignifies them, and it shouldn't be done. I mean, I was asked to debate an evolutionary biologist a while ago about the origins of COVID, and I didn't, because it dignifies them by putting someone up with credibility against them. So no, I'd be happy to go on a podcast and talk to people, but debating, no. Thank you, Mark, for your contribution. I appreciate it, and thank you, Mario. Buon anno, dottore e famiglia. Chin chin, thank you. Anche a lei. Are mRNA vaccines the best we have ever had? Well, if you measure best by percent, the effectiveness, yeah, they're really good. My vaccinated daughter spent the evening with my unvaccinated daughter. Next day had symptoms and tested positive. Unvaccinated, did not catch the virus. Any thoughts? It's kind of soon, right, for that to be the source. It's a very quick incubation period of 1 day. And you have to try and rule out other sources, right? This is always the case when you say, this must have happened here. But often there's some other source of the infection. Thank you, Ken, for your contribution. I'm glad you like it. We're happy to help. Not everyone agrees with us, but we're here to provide information to the best of our ability. And as I said, I've been working on viruses and thinking about them for a long time. And yeah, we did some rapid fire answering. This is the way we usually do. I'm sorry that I can't answer more of your questions. I wish I could. It would just take hours and hours, and I don't want to keep you all here. But before we wrap up, I have about 900 people here. 550 you have given a thumbs up. Please, more of you give it a thumbs up there. It helps. I just want us to be seen by more people. And Tom points out this is an enormous problem in science communication, the role of credentialed, self-styled contrarians, deniers, cranks of uncertain intent. Yes, people like to be heard, and people think they're credentialed, like cardiologists, right? I just have a thing about that cardiologist. He doesn't know what he's talking about. Thank you, John, for your contribution. Thank you, Thomas, for your contribution as well, supporting science education, right? And if you didn't get your question answered, I'm really sorry. Come back next week. I'll be here with Amy again next year, 2022. This is the last one for 2021. Good riddance. Thank you, Michael, for your support. Lots of great questions. Bring them back next week. Thank you, Hoplite76. Omni immunity protects against Delta, and if there's no Omni reinfection, short of a new variant, will the pandemic end? Well, the pandemic is going to end no matter what, because the 1918 pandemic ended without a vaccine, right? There you go, cosmic lettuce. It ended by enough people getting infected that there was enough immunity. And this will end sooner because we have vaccines that work. So I don't think we've seen the last of the variants, but I'm not worried. Throw them at me. We have good vaccines. They work. When they stop preventing a disease, serious disease and hospitalization, then we can change them. Thank you, I Canada, for your support. Took your 2020 course when COVID first started. My pleasure. Good virology movie for entertainment. Contagion is probably the best. And go watch it and tell me 3 things that are wrong with it. And you can come back next time. Thank you, Susan, for your contribution. I try to be a voice of reason, even though I get a little excited, right? I can't avoid that. I got that Italian blood in me. Thank you, Kat. Not blood, but genes, right? Appreciate everyone's support. Thank you, Stephen. Great support tonight. Really love how everyone is into this communication that we're trying to do. Roosby, thank you so much. Is a year after the second RNA too long spacing? No, it's fine. The real issue is too short. A year will be fine. Go for it. Mystery Fine, thank you so much. My homework is to go back and watch all your virology lessons. Please do. The most recent one is Virology Live, which I live streamed. The course ended today. Sniff, sniff. I'm so sad it's over. 25 lectures. And you can see people asking great questions because there's a chat there just like this. You don't have to do it in a short time. You can take your time. Please, the more people that listen, the better off the world is going to be. Thank you, New York Cohen. 3 times mRNA vaccinated person, slightest line ever on an antigen a week after first testing positive. Good to go back to normal. Yes. Slight ever, even though these antigen tests are not quantitative, you're 3 times vaccinated. You are fine. You are absolutely fine. Thank you, German opera singer, for your support. Really appreciate it, too. And I can't tell you how it pains me to scroll through all these questions. Thank you, Michael, for your contribution. Thank you, Datebayo, for your contributions. What do vaccinated parents' families do if they have children under 5 in the household? Should we be worried that we will bring COVID home? Yes. Even though children are less frequently infected, they can be infected. They can die. You have to be careful, for sure. You have to wear a mask and be careful among the kids until the time that they can get vaccinated, for sure. Thank you, Vincent, for your support. Really appreciate it. And I'm almost done here. All right, I'm down at the bottom. And I apologize for scrolling through all your questions, but there are a lot tonight. There are 900 people, and if I took one from each, it would still take hours and hours. So I hope you learned something tonight. Thank you all for coming. Thank you for your support. Thank the moderators. Kind of a thankless job. Vanity Nutrition, Mr. Ozzy Steph, and Les. Appreciate your help all throughout 2021. And here's to a 2022 with good science information and a receding pandemic. Happy New Year, everybody. Be safe, and we will see you next year. Good night.